So class, in this recording, we're going to focus on glycolysis, the first and primary step of cellular respiration. This step can be summarized in a sentence, and I gave you, the bottom line. Glycolysis is going to break glucose molecules down into small pieces, and these small pieces are then going to be sent into the mitochondria of the cell. As we look at glycolysis, it's also worth emphasizing glycolysis happens in both anaerobic and aerobic respiration. During glycolysis, we're going to have a glucose molecule being dumped into the cell, and we need to kickstart the chemical reaction. And to kickstart the chemical reaction, we need to add some more Gibbs free energy in the form of ATP being expended. So we need to raise the energy level. And to raise the energy level, we're going to burn through not one, but two glucose molecules. This is kind of like the spark you need to start a fire, or the striking a match. And if you think of a match, matches have lots of chemical energy stored in them. But the chemical energy in the match is fairly stable. It's not released until you supply some activation energy in the form of heat, frictional heat, when you strike it on the sandpaper of the box. Now, when we think of these chemical reactions of glycolysis, glucose has chemical energy in it. We need to get that chemical energy out. But in order to get the chemical energy out, we first have to raise the energy state. We supply the activation energy. And then once we've raised the overall energy of the molecule, then we can start to break it down into smaller pieces and reduce the energy level. At the very end, one glucose is going to be turned into two pyruvic acids. And it's those two smaller pyruvic acids that can be processed and sent into the mitochondria. Since we have a net loss of energy levels start here and they end down here, so we have a net loss of energy. If we look at the y-axis, we refer to this reaction as exergonic. It also is going to be referred to as catabolic because we start with one larger molecule and end up with two smaller molecules. So as we look at glycolysis, we need to emphasize some key take-homes. First, it does not require oxygen. Glycolysis can happen with or without oxygen being present. It also happens in the cytosol of the cell, not in the mitochondria. The net equation for glycolysis is right here. You do not need to commit this to memory, but I do want to emphasize some key ideas here. We start with the glucose. We end with two pyruvic acids. So the glucose is turned into two pyruvic acids. As we look at the NADs, we take two NADs and convert them into two NADHs. So we have two high energy electrons that are going to be carried away. And we'll come back to those in a bit. We can't forget about these high energy electrons. They'll be a key player later on. And also, we have a net yield of 2 ATP. We use 2 ATP to kickstart the reaction, but then we produce 4 ATP. So if we have a gross of 4 and we used up 2, our net yield would then be 2 ATP. To give you an idea of the overall complexity of this, we start with glucose up here on the top, and you do not need to memorize all of these intermediate species that exist. But I want to focus on how we have glucose at the top. Glucose is then going to be split in half to make two pyruvic acids. And notice how we used up 2 ATP here, so we expended 2, but then we gained 4 down here to give us that net yield of 2 ATP with glycolysis. Now, as we're looking at the glycolysis, I had mentioned briefly earlier that it can happen with oxygen or without oxygen present. When we have that oxygen present, we refer to that as an aerobic chemical reaction. And this is our preferred mode of making ATP because we make much more ATP when we have oxygen present. This is a much more efficient mechanism of making cellular energy. Also, I mean, we like to breathe. I don't know about you, but I really like breathing. It feels good to breathe. And then when we look at anaerobic respiration, anaerobic respiration means that there is no oxygen, or when oxygen is not present, so without oxygen. And this anaerobic respiration can make some ATP. It depends exclusively on glycolysis. So instead of getting lots of ATP per glucose molecule, we get only 2 ATP per glucose molecule. It makes a lot less ATP than aerobic respiration does. As we look at anaerobic, and this is the natural point in the discussion where we need to take a little side tangent and talk about anaerobic respiration. We'll come back to aerobic in a bit. When we think of anaerobic respiration, if we have oxygen available, that pyruvic acid or pyruvate is going to be sent to the mitochondria and processed to make lots and lots of ATP. But if we don't have any oxygen, or if we think of like a red blood cell, for example, red blood cells typically have lots of oxygen, but they don't have any mitochondria. The cells themselves can't generate ATP through aerobic respiration, and they instead only are going to generate ATP through glycolysis, that first part of cellular respiration. And when we think of glycolysis, I had shown you in a couple slides, 3 slides ago, how we have an NAD positive carry away a high energy electron. But if we can't regenerate these NAD positives, we can't continue this process of glycolysis. And when we look at anaerobic respiration, we are going to take that high energy electron in the form of an NADH, and that high energy electron will then be donated to a pyruvic acid, and we take that pyruvic acid at the end and turn it into a lactic acid. And this process of turning pyruvic acid into lactic acid regenerates the electron carriers and allows glycolysis to continue to occur. The downside of this is that we build up lots of lactic acid inside of the cytosol of our cells. So as we have lactic acid, or lactate, potato, build up inside of our cells, we need to get rid of it. And we get rid of it by dumping it into the bloodstream, and from the bloodstream, it makes its way over to the liver. While that lactic acid is in the liver is going to oxidize it, which is a way of saying that it strips electrons away from it. And as it's being oxidized, it's turned back into pyruvate. This lactic acid takes a lot of oxygen to be processed. And this is one of the reasons why we experience oxygen deficit. When when people go for a run or spend a lot of time exercising and really exert ourselves, we build up lactic acid in our bodies, and then when we're done exercising, we start huffing and puffing. We have to breathe really hard for a while. And the reason we're breathing really hard is because we had to take all of this lactate that's built up in our bodies and use up oxygen to turn it back into pyruvic acid in our livers. This is our post-exercise oxygen consumption. And there are other reasons why we have oxygen deficits as well. We have to replenish the myoglobin within our skeletal muscle tissue. That's another notable reason. Now, as we're looking at this process of anaerobic respiration, this lactate can also be, in addition to being turned into pyruvate, can be turned into glucose 6-phosphate. This is just a glucose that has an extra phosphate molecule attached to it onto the 6th carbon. And that glucose 6-phosphate can then be turned into glycogen and stored in the liver as a medium term or a form of glucose that has medium availability. And when we look at this process, this glycogen can be turned back into glucose by removing the phosphate groups from them, and that free glucose will then be dumped back into the bloodstream. A lot of students miss the key point here, that this only happens in the liver. This does not happen with other tissues of our body. The liver is the only organ in the body that will dump glucose into the bloodstream, that will store glucose and then dump glucose in the bloodstream. Now, when we think of anaerobic respiration, and there's some pros and cons. The pro of anaerobic respiration is that we can still make make ATP out of glucose when we aren't getting enough oxygen. But a drawback is that it's kind of wasteful. Most of the energy to glucose is still stored in that lactic molecule and really hasn't done anything useful. And then as we have lactate built up in our body, lactate alters our body's pH. Lactate breaks homeostasis. Lactate is toxic to us. We also find that our skeletal muscle tissues are fairly tolerant of anaerobic fermentation. This is one of the reasons why when you're exercising, you can sometimes, we call it the lactic acid burn. And ironically, it's not directly because of the lactic acid, but skeletal muscle tissue tends to build up a lot of lactic acid within it during anaerobic respiration. Cardiomyocytes, or cardiac muscle tissue, is less tolerant of anaerobic respiration. Our heart is designed to prefer that oxygen, and we need a very good oxygen supply to our heart. Here's something else that's worth emphasizing. Our brain does not employ any anaerobic fermentation. Our brain gets preference when it comes to our oxygen supply. More than any other organ in the body, our brain needs a high supply of glucose and a high supply of oxygen because the brain uses up tons and tons and tons of ATP. This is one of the reasons why if somebody is having circulation cut off to their head for just a few minutes, they can have brain damage. I remember when I was in middle school. Yeah, I was in middle school. We would play around in the locker room and put chokeholds on ourselves. And in just as little as 10 or 15 seconds, if you put somebody in a sleeper hold, you can knock them out and they would pass out because of the lack of blood going to the brain. And in just 10 or 15 seconds, your brain can go from conscious to unconscious. That's how sensitive our brain is to a lack of oxygen and a lack of ATP. So as we're looking at aerobic respiration, we're done with our tangent. Let's go back to tangent of anaerobic respiration. Let's go back to the primary show, which is aerobic respiration. So we're going to assume we have plenty of oxygen. And when we have lots of oxygen, that pyruvic acid is going to go from the cytosol and ultimately be processed to be brought into the mitochondria. And this is where we remove the first carbon. So remember, we started with one glucose molecule. That one glucose molecule got converted to pyruvic acids, or pyruvates, and then we take this CO2 functional group, this carboxylic acid functional group off of the pyruvate, and then this carboxylic acid functional group is cleaved off and turned into a carbon dioxide. And then this leftover segment is then going to have a high energy electron stripped off of it, and then bound to coenzyme A to be turned into acetyl-CoA. And this acetyl-CoA is what we are going to be directly dumping into the next phase, which is the citric acid cycle. So if you have any questions or comments about glycolysis, or if you have any questions or comments about anaerobic respiration, please feel free to post them on the discussion board or to shoot me an email. And as always, class, happy studies.